Assalamu alaikum and good morning to our course instructor Ishra Chan ma'am and my fellow classmates. Welcome to our presentation titled as Database Searching FASTA. Okay. These are my teammates and these are the outline of our presentation such as database searching, real life examples and scenarios, sensitivity, selectivity and precision, how FASTA algorithm works, building query table, hash table, target table and expected tables, building offset table, pre-final table, final table and conclusion. Okay, I am MD Shafawat Islam and my ID is 262159121. Let's start my presentation with the basic of database searching. At first, we all know about what is model evaluation. Model evaluation is the process of measuring how well a computational model performs on biological data using different evaluation metrics. And these are the metrics like accuracy, sensitivity, or recall, specificity, precision, confusion metrics like TP, TL, FP, FN. And this is the diagram or figure of evaluating something. It is the picture of evaluating model of data. Let's talk about why model evaluation is important. Model evaluation is important because it checks how accurate and reliable a model is. It helps researchers compare different models, reduce prediction errors, and make better decisions in biological research, disease diagnosis and treatment planning. Here is two diagrams or two figures that ensure how or why model evaluation work, and they are searching and comparing something. Okay, let's talk about confusion matrix. At first, we all know about what is confusion matrix. A confusion matrix is a table used to evaluate the performance of a classification model. It compares the actual outcomes with the predicted outcomes to show how well a model is performing. Okay. It is commonly used in machine learning, medical diagnosis and fraud detection to measure accuracy, precision, recall and other performance metrics. And these are the key points or keywords. How it works or why it's important. The first one is compares actual versus predicted results. And next, helps identify correct and incorrect predictions. Contains 4 outcomes: TP, TL, FP, FN. That's positive and negative, false positive, false negative, true positive, true negative. And last one is used to calculate accuracy, precision, recall, specificity and FOR score. And it is a symbol or a sign that ensures the accuracy and true positive. And the last one is why confusion matrix is used or why it is important. A confusion matrix is used to understand how well a classification model performs. It compares the actual results with the predicted results and shows where the model makes correct and incorrect predictions. It helps identify different types of errors such as false positives and false negatives. This information is useful for improving the model and making more accurate predictions. And what is the application of confusion matrix? And here is the point. And first one is disease diagnosis, cancer detection, spam email filtering, fraud detection, image classification. Okay, so we can say a confusion matrix is very important for identify between or compare between the right decisions with the predicted decisions. Thank you everyone for my presentation. Now I would like to hand out my next presentator, MD Sharif. He will present the next step. Thank you.
Good morning, respected teachers ma'am and following my fellow classmate. My name is Mutashimullah Sharif. My ID is 262-15850. And following Shafqat's excellent introduction to model evaluation and confusion matrix, I will be walking through a real life medical example to illustrate the diagnosis scenario. Change the slide. Let's ground the concept in practical high stake scenario. Imagine there is a patient who has figured that he has cancer and goes to the doctor for diagnosis. And in computational biology and medicine, the accuracy of diagnosis is everything. As the slide note adapted from the voluntary. The art of medicine consists of amazing the patient while natural cure the disease. But the science of medicine rely on getting the diagnosis right. That means proper diagnosis is everything. Next slide. Let's look what happened when the diagnosis model are right. There are two types of prediction. One is true positive and another is true negative. True positive means when the patient has cancer and machine detected actually cancer and machine give positive sign. It means true positive. And true negative is the alternate thing where the patient has no cancer but the diagnosis came negative. That means patient have no cancer but diagnosis came negative. That is true negative. And the famous statistician George B.P. Box said that all models are wrong but some are useful. This quote is related to this true form. Let's come to next slide. Next, there is another scenario that is incorrect prediction. Incorrect prediction means there is also two scenario that is false positive, false negative. False positive means patient have no cancer, didn't have cancer, but machine came with positive. Positive, that means patient is safe. But when another thing is very dangerous for a patient because that patient had cancer, but diagnosis came in negative, and the patient didn't know that he have cancer, and he will be treated like a normal person. So he don't get any proper treatment for his diseases. So it is a very risky scenario overall for all. So ultimately in this in God we trust, and all other must bring data. Let's jump to next slide. In summary, there is a four scenario in summary. That is the main safest zone is true positive, and the most dangerous part is true negative. And approximate answer to right problem is would be a good deal more than an exact answer or approximate problem.
Assalamu everyone, I am Swati ID 2621589. Thanks to my previous presenter Sharif for his wonderful presentation. Today I will be presenting on a crucial topic in biological data analysis, sensitivity and selectivity. We will cover what this term means, look at their formulas, and walk through some practical example to see how they work in real world through biological context. Let's start with sensitivity, and also known as recall. In simple terms, it measures a test's ability to correctly identify positive intentions, such as individuals who actually have a disease. When we say when we say a test has high sensitivity, it means it rarely misses any true positive case, thereby minimizing false negatives. In biological context, we use this to evaluate how effectively a classifier finds or actual target sequence character. For example, finding FBC residui in a DNA sequence. Now let's move to the second slide. Calculate this, we use the formula sensitivity is equal to true positive by plus false negatives. We see this through a practical example. Imagine a scenario test given to 100 patients who actually have a disease. Here, the machine truly identified 90 of them. These are our true positive. However, 10 are missed. These are our false negative. And through the formula, if we apply this, sensitivity is equal to 90 plus 10 is equal to 90%. This means the test correctly identified 90% of real positive case. Move the next slide. We move to specificity and selectivity. This refers to a test ability to identify positive claims without including non-target intents, which minimize false positive. High specificity confirms that when a target is detected, it is truly genuine and not just a false alarm. In sequencers, we focus on measuring what function of all detected characters are actually the character. Now let's move to the third slide. Next. The formula for selectivity is true positive by true positive plus false positive. These are our. Let's apply to a bioinformatics example. Suppose a tool scans a short FASTA DNA sequence to find cytosine bases. It flags some base and 3 of them are actually C. But one base was actually Z, which tool mistakenly identified as C. The calculation is 3 by 3 plus 1 is equal to 75%. The algorithm has a 70% selectivity or precision. This means that when it predicts a base is C, it is correctly 70% of the time. To summarize this, let's compare precision and recall. First, talk about recall. Recall measures how many of the actual positive cases are successfully identified. It minimizes false negative and prioritizes coverage or completeness. If we say this in simple terms, essentially quantity over quality. If we talk about precision, it measures how many of the predicted positive causes are actually correct. It minimizes false positive and prioritizes quality and exactness, or quality over quantity. Both are essential for ensuring high prediction accuracy and quality in biological models. Conclude my presentation. Thank you all for your attention.
Assalamualaikum everyone. My name is Jaina Kartuma. My student ID is 262158225. First of all, I would like to thank our previous speaker Shodip Hussain. Today I will discuss about FASTA algorithm. First question is, what is FASTA? FASTA stands for Fast Alignment Chart Store Cake. FASTA is the bioinformatics sequence alignment algorithm used to compare a DNA RNA protein sequence with sequence storage in database. It finds reason of similarity between sequences with help scientists identify region, proteins and possible bioinformatics function. Right. How the FASTA algorithm works? Input the query sequence DNA, RNA or protein, break the query sequence into one, scan the database for sequence containing the same one, identify the region with many matching one, join nearby matching region to create longer alignment candidates. Far from database alignment, score the measured similarity. Display the best matching sequence as the final result. Next slide is application of FASTA. Phylogenetics, genomic research, proteomics and biomedical studies. Phylogenetics, evolutionary studies. Genomic research, DNA RNA sequence compare. Proteomics, protein structures analysis. Biological studies, study of disease gene identification. Next slide is advantage of FASTA. FASTA sequence charging compared to method. Work with DNA and DNA, RNA or protein sequence. Easy to use and widely accepted in bioinformatics. Help identify homologous genes and proteins. Thank you everyone.
Assalamualaikum everyone. I hope you all know me. I am Muhammad Abir Hassan and my ID is 262158889. So in this part of our presentation, I will practically show you how we can build a query table, a hash table using query sequence, a target table and an extended target table using the hash table and query table. So without any further delay, let's jump to our topics. So first of all, to build a query table, we have to take a look to the query sequence. Here our query sequence is just this link, as you can see. So first of all, we will take each letter from the query sequence and put it in the table in a manner like 1, 2, 3, 4, 5, 6, 7. Just like that. It was very easy. So the most interesting part is the hash table. So to make hash table, first of all, we have to take the query sequence and lexicographically manner them to put in the hash table. Like the first letter of English alphabet is A. So there is A in our query sequence. So we will first of all take that A and then we will take C. Why C not B? Because there is no B in our query sequence. Just like that, we will lexicographically manner the alphabets to the hash table. And then for A, as you can see that A is at the 10th position. So we have taken A and put it in the 10th position. And then you can see that C is at the 6th position. So we have taken C to the 6th position. And after that, you can see that A is at 3 positions. First of all, 7, then 9, and then 13. So we have taken its 3 positions, 7, 9, and 13. Just like that, we will continue to make. And last, I will say that the letter U is in 2 positions, in 2 and 12. So we have put 2 and 12 for U. So that was very easy to make the hash table for query sequence. Next slide please. So here we have to make the target table. So target table is just similar and just like the query table. So we will take the target sequence and make a box where we will take the numbers in manner and then we will just put the target sequence in the target table. Next please. So here is the most important and most interesting part of this part of the presentation. So first of all, to compare these two boxes, we have to take them in the same position. So we have just taken the two tables in the same position, so we can compare both of them. Next slide please. So we will make the extended target table based on the hash table. In the previous slide, we have taken the two tables in the same slide. Here is the hash table for query sequence and here is the target table. So to make the extended target table, this part we have to take a look at the extended target table and the target. So here the first letter of the target table, as you can see, that is L. So we have to find out where is the L in the hash table. So the L is at the 8th position. So our rule says that we have to take the position of the letter in the hash table and then we have to subtract that from the position of the letter in the extended target table. So here the position of L is 8 and here is 1. So 8 minus 1 is equal to 7. And then we will see that E. So in the target table, the E is at the 2nd position and in the hash table, it is in 3 positions. So we will take one by one. Like first of all, 7. So 7 minus 2 is 5. Then 9 minus 2 is 7. And at last 13 minus 2 is 11. It was easy, right? So then we will take A. So the letter A is at the first position of the hash table. So we will take 10 and minus 3 from 10. So we will get 7. And then we will take E. So U is at the fifth position in the target table, and in the hash table it is at two position, 2 and 12. So we will minus 2, 5 is equal to minus 3, and 12 minus 5 is 7. And then as you can see that these boxes are empty. Why? These are not. Why this happened? Because if we focus on the the hash table, you will see that there is no O, A or N in the hash table. So there is no position of O, F, N in the hash table, so that we can subtract from that. So we will not take them and we will keep the boxes empty or null. Thank you for your attention, and I hope you understand. Thank you everyone. So I will now welcome my next presenter, Sabir Hussain, to continue the presentation. Assalamu alaikum.
Assalamu alaikum everyone, and my respected ma'am, Ishachan ma'am. I am Sabir Hussain, my ID is 262155855. Now I will be taking you through the final matching phase of the FASTA algorithm to conclude our presentation today. After my teammates successfully built the extended target table, our next crucial step is constructing the offset table. So, next slide please. Here we see that a new table that ranges sequentially from the minimum entry to the maximum entry, found in the extended target table. Directly beneath each of these entry numbers, we write down the exact number of times and a specific entry occurred. For example, looking at our data, that entry 7 occurred 6 times, which is highly significant, whereas the entry 1 is only 2 times. Once the offset frequencies are mapped, we build the prefiner table. Next slide please. In the prefiner table, we initially align both the query sequence and target sequence starting from the position of 0. Now we move on to building the finer table. Next slide please. In that finer table, which is the where optimal alignment is logged in, we look back at our offset table to find out which entry numbers occurred the maximum number of times. As we noted, in this case it is 7, which occurred 6 times. To determine exactly where is the sequences align best, we add the maximum entry number to the previous starting position of the target sequences. Since our initial starting position was 0, our new starting position from the target sequence becomes 0 plus 7. This simple mathematical shift represents the region of highest similarity between the biological sequences to conclude our presentation. Next slide please. In the conclusion, this presentation demonstrates how performance evaluation metrics such as precision, recall, and confusion matrices integrated with computer sciences data structure such as hash tables and offset to perform fast biological sequences elements. By computing positions offset through hash tables rather than checking every character combinations manually, the FASTA algorithm significantly reduces computation time, making large-scale genomic and protein database searches efficient. Thank you all. Thank you, ma'am, for your patience. And this was my presentation.